Hi, this is Dr. O'Connor, and in this video we're going to talk about generation of biochemical energy. We have to talk about that and about some chemical reactions, and then we're going to get into the citric acid cycle and the electron transport chain. In the next video I'll talk about carbohydrate metabolism. So let's go ahead and move on. We all know that living organisms must do chemical work. And what is that chemical work? Well, we have to synthesize biomolecules that are needed for energy storage, growth, repair, and replacement. Cells need energy in order to move ions and molecules across cell membranes. Remember, we need to get nutrients into the cell and waste out of the cell. So the energy that's released from food will allow the above work to be done. So some energy requirements. Energy must be released from food gradually. If it was released all at once, well, we'd have maybe a little explosion there, right? We can't do that. So it has to be released gradually. It must be stored in forms that are accessible. The release of energy from storage has to be controlled so that the energy is available exactly when and where it is needed. And only enough energy must be released as heat in order to maintain body temperature. Energy in a form other than heat must be available to drive chemical reactions that are not favorable at physiological temperatures. I'm going to talk to you a little bit about spontaneous reactions. And we haven't talked about them before. We haven't talked about spontaneity, but basically a spontaneous reaction is one that's going to proceed without any external influence. So when I turn on the gas on my stovetop, there's a little spark, okay? That gets the reaction going between the gas and the oxygen, right, to produce the heat we need to cook our food. And as long as I don't run out of gas or oxygen, that will continue. That reaction will continue without any external influence. This quantity here, this is what we call free energy. It's actually Gibbs free energy. And we see that it's dependent on enthalpy. And we did not talk about delta S. And we won't talk about it here. We don't need to. But all you need to know is that this delta G, this refers to energy that is released from a chemical reaction in order to do work, and that is available to do work, or energy that is needed in order for the chemical reaction to take place. So we describe favorable reactions as being exergonic, and these exergonic reactions release free energy. And these are the source of our biochemical energy. So again, as I said, exergonic applies to the release of free energy. That free energy then is available to do work. And we represent that by a negative delta G, okay? And don't mix up exothermic and exergonic. Exothermic, that applies to the release of heat only, whereas delta G, a negative delta G, or an exergonic reaction, that is dependent on both delta S and this other quantity called entropy. Okay, so that is, exergonic refers to the release of free energy. Exothermic, that refers to the release of heat. So let's go back up here. I have these two graphs here. Notice I have free energy versus the progress of the reaction. And we've seen these before, but instead of free energy, it was potential energy, okay? And remember, the difference between the energy of the reactants and products was delta H. But this is free energy versus reaction products. And the difference between the reactant and product energy is delta G. And in this case, we have an exergonic reaction, meaning heat is released. So delta G would be negative. In this one, here we have our delta G, and the energy of the products is greater than that of the reactants. So therefore, delta G is greater than 0. So that means that, number one, the reaction is unfavorable, okay? And our products have more energy than the reactants. So when delta G is negative, then we have a favorable reaction. And that means the products have less energy than the reactants. So that's, I do want you to know this. So for example, if we look at photosynthesis, here we have carbon dioxide and water. So this is photosynthesis going to the right. And the product here is glucose and oxygen. But look at this. Delta G is positive. So that means that it's an unfavorable reaction, right? That's unfavorable. And it's endergonic. That means energy is required. Well, where does that energy come from? It comes from the sun, okay? And then let's take a look at the reverse, okay, of photosynthesis. That's actually an oxidation. Notice the delta G has the same magnitude, but it's the sign that's different. So the reverse is negative, meaning the reaction is spontaneous or exergonic, if you will. In other words, energy is released. So when you have a reaction that is spontaneous in one direction, it is going to be non-spontaneous in the reverse direction, or vice versa. So let's go ahead and take a look at, here we have some cell components and their principal function. You can look up the diagrams in the book, but you need to know some of this. Okay, so here's a diagram of the cell. And so what we have is the, here we have the cilia, and that's important for movement of materials like mucus from the lungs, okay? We have the Golgi apparatus, okay, and that's going to be right here. And that is the synthesis and packaging of secretions in the cell membrane. And then we have the mitochondrion, okay, and this is right here. And this is where the synthesis of ATP from ADP takes place, okay. So the mitochondria is often called the cell's power plants, and they're one of the most important of the organelles for energy production. So it's in the mitochondria that about 90% of the body's energy-carrying molecule, ATP, is produced. So that's very important. Okay, so the mitochondrion is roughly an egg-shaped structure, and is composed of a smooth outer membrane and a folded inner membrane. The space enclosed by the inner membrane is called the mitochondrial matrix. Okay, so that'd be the space enclosed. And it's in that matrix that the citric acid cycle and the production of most of the body's adenosine triphosphate, or ATP, takes place. And there are coenzymes and proteins that are involved with this. We haven't talked about enzymes, but enzymes, many of them are proteins, and incorporated in some of these proteins are, it could be molecules like heme, we talked about heme, or it could be metals. And enzymes are biological catalysts. So many of our biochemical reactions require enzymes in order to speed up the reaction. Remember, a catalyst speeds up a reaction without itself being consumed. Okay, so we talked, let's see, about the mitochondria. Here, let's see, what do we have here? Here we have the nucleus, okay, of the cell. And this is where DNA replication takes place. And then we have, let's see, after the nucleus, we have the rough endoplasmic reticulum. Okay, so that'll be here. and that's where protein synthesis and protein transport take place. We have the ribosomes, that is shown right here. And the ribosomes, that is where protein synthesis takes place. Okay. Then we have the microvilli. Okay, that's these little hair-like structures here. And absorption of extracellular substances, for example, in the digestive tract. Okay, that's where that's important for that. We have the cytosol, which is right here. And the cytosol, this is intracellular fluid, and it contains a lot of dissolved proteins and nutrients. And then we have the lysosomes represented here, and they remove pathogens or damaged organelles. The smooth endoplasmic reticulum, we see that right here, and this is where lipid and carbohydrate synthesis takes place. And then we have this shows the cell membrane, and of course this is composed of lipids and proteins that govern entry and exit from the cell and delivers signals to the interior of the cell. So take a look at the diagram in your book so that you know what's going on with the cell. So now let's talk about metabolic reactions. Let's go ahead and define metabolism. These are all the chemical reactions that take place in a living organism. Catabolism, these are metabolic reaction pathways that break down food molecules and release biochemical energy. Anabolism, these are metabolic reactions that build larger biological molecules from smaller pieces. That includes those that store energy. So you can think of it this way. Catabolism is the breakdown of larger molecules to smaller molecules and, of course, energy. Whereas anabolism is the building up of smaller molecules to larger molecules. Together, all of the chemical reactions that take place in an organism constitute its metabolism. We'll see in a little while that food molecules undergo catabolism in order to provide energy in 4 stages, but I'll show you that in a minute. For now, what I want to do is talk about metabolic pathways. So, as we said before, all of the chemical reactions that take place in an organism constitute that organism's metabolism. So, metabolic pathways can be linear, cyclic, or spiral. Okay? So here's an example of a linear sequence. So you have, this reaction going from A to B, and then in order to produce C, you have to have B, okay, and so on. And each one, of course, is catalyzed by an enzyme, usually a different enzyme. So this would be a linear sequence. A cyclic sequence, we're going to talk about one of those later, the citric acid cycle. And in a cyclic sequence, what happens is there are several steps, and the product in the last step, the reactant in the first step of the next round of the cycle. And then we have spiral sequences. And I'll show you that a little bit later when we get to fatty acid oxidation. If I explain to you what it is now, you're going to forget by the time we get there. So here are those 4 stages that I was talking about. So here we have digestion, catabolism, and energy production. So this all starts off with food, and it's either going to be lipids, carbohydrates, or proteins. And in step 1, we have digestion of these lipids, proteins, and carbohydrates. So basically, enzymes in the saliva, stomach, and small intestine convert these large food molecules into smaller molecules. The smaller molecules then, we have our fatty acids, glucose, our amino acids from the protein. These are transferred to the blood for transport to cells in the body. Now, in stage 2, what we have is small molecules from digestion are going to follow separate pathways that move their carbon atoms to 2 carbon acetyl groups, okay? So, for example, our fatty acids and glycerol will undergo fatty acid oxidation, and here we have acetyl coenzyme A that is produced. Glucose and other sugars undergo glycolysis to pyruvate, and then from pyruvate to acetyl coenzyme A. And then the amino acids from the proteins will undergo amino acid catabolism and go into the citric acid cycle. So acetyl coenzyme A, which I'll talk about in a minute, is the molecule that is going to go into the citric acid cycle. So in step 2, we have acetyl coenzyme A production. So our sugars, our fatty acids, and our amino acid molecules are degraded further in order to yield acetyl coenzyme A. In stage 3, we have the citric acid cycle. So acetyl coenzyme A is oxidized, and this takes place in the mitochondria, and it's oxidized in the citric acids by the citric acid cycle to yield carbon dioxide, some reduced coenzymes, and ATP. And then finally, stage 4, this is the production of ATP, or high energy molecule. And so the energy transferred to the reduced coenzymes in stage 3, okay, citric acid cycle, this is used to make ATP by the pathways, the electron transport and oxidative phosphorylation pathways, which are coupled. I'll talk to you about what it means to be coupled in a little while. So those are the 4 main stages. So we'll take each one and talk a little more in detail as we go. So let's take a look. What is this acetyl coenzyme A? Well, first off, what is an acetyl group? An acetyl group has 2 carbons, and there's an oxygen bonded to carbonyl carbon. So this is the acetyl group. So the acetyl group attaches to the coenzyme A, okay? And that's through this sulfur bond. So there's a sulfur and the coenzyme A, which I can show you right here. All right, so this is the coenzyme A molecule. Notice we have a thiol group here, the SH. So what's going to happen is the acetyl group is going to bond to the coenzyme A with the carbonyl carbon bonding to that sulfur atom. Okay, and then it's the coenzyme A then that carries the acetyl groups to the citric acid cycle. So this is an intermediate in the breakdown of all classes of food molecules, lipids, carbohydrates, and proteins. I will say right now, coenzyme A is neither an oxidizing nor a reducing agent, so it doesn't work that way. So the next molecule that we want to look at is ATP, adenosine triphosphate. Oh, tri, it must have 3 phosphate groups. And it does. It has 3 phosphoryl groups, okay, which are these groups here. And these phosphoryl groups, when one of these phosphoryl groups is removed, then we get adenosine diphosphate, which is ADP. Well, it turns out that when one phosphoryl group is removed, we end up with ADP. So energy is released when a phosphoryl group is removed. So we see that here. We have ATP being converted to ADP. Notice here the removal of that phosphoryl group. And notice our delta G, or the energy released, is 7.3 kilocalories. Okay? And so this would be an exergonic reaction. It turns out that the biochemical energy that's needed is gathered from exergonic reactions that produce ATP. Okay? ATP is available where energy is needed, of course. ATP hydrolysis releases the energy for whatever work needs to take place. So, in other words, the removal of that phosphoryl group, that's a hydrolysis reaction. A lot of your book says that the energy is stored in the bonds, and that's not quite true. The energy is released because of that hydrolysis reaction. It is the hydrolysis that is exothermic. Okay? So that hydrolysis reaction releases the energy for the work that needs to be done. Energetically unfavorable reaction can be coupled with an energetically favorable reaction. Remember, not all reactions are going to be favorable. All right, many are, but not a lot of them aren't. But basically, when these reactions are coupled, what we end up with is an overall reaction that is favorable as far as the free energy change. As far as the rate of ATP hydrolysis, it is slow. So, there's no catalyst present. So therefore, the stored energy is released only in the presence of the appropriate enzyme. I have a chart. It just shows some free energies of some of these phosphate compounds when they undergo hydrolysis, okay? But we see they're all negative, okay? Phosphoenolpyruvate actually releases the greater amount of energy. But anyway, back to couple reactions. These are pairs of reactions that occur together. The energy released by one reaction is absorbed by the other reaction. So coupling an energetically unfavorable reaction with one that releases more energy than the amount required is very common in biological reactions. And we'll see that over and over again. I want you to know that coupled reactions do not take place separately. The net change occurs as represented by an overall reaction. So here, notice that the formation of ATP is coupled with the hydrolysis of phosphoenolpyruvate. And this has got a higher energy, or a higher free energy, than the ATP, okay? So this is a negative 14.8, okay? Here, when we have ADP and we want to add that phosphoryl group to it, this is going to need 7.3 kilocalories of energy. So when these two reactions are coupled, what we can do is we add the two reactions, we add the free energies here, and we end up with a reaction that is favorable. In other words, it has a negative delta G. It's exergonic. So here we have our phosphoenolpyruvate, our ADP, and this phosphoryl group is transferred to the ADP to form ATP, and this turns out that it's an exergonic reaction. We coupled the reactions are coupled, and this results in an overall favorable reaction. So again, as I said, the net change occurs by the overall reaction, okay? Sometimes in your book or another book, you're going to see this type of notation. You're going to see this curved arrow here. So on the left side of the curve, this just means that ADP is a reactant here. On the right side, that means ATP is the product. So just like it is here, just like ADP is the reactant here. So that's what that means. So what we want to do is we want to talk a little bit about oxidized and reduced coenzymes. And we know that the net result of catabolism is the oxidation of food in order to release energy. Many of our metabolic reactions are redox reactions. So just to refresh your memory, oxidation we think of as the loss of electrons. It can be an increase in oxidation number. And we also look at it as a loss of hydrogen or a gain of oxygen, okay? So reduction, on the other hand, can be a gain of electrons or a decrease in oxidation number, gain of hydrogen or loss of oxygen. And we know that oxidation and reduction always occur together. You can't have one without the other. So when we're going down here we start with methane. And notice we've increased the number of carbon-oxygen bonds. So this here is an oxidation, or this substance here has been oxidized to this one. Notice as we go even further, notice again the increase in the carbon-oxygen bonds. This is an oxidation. Notice while the carbon-oxygen bonds are increasing, the number of carbon-hydrogen bonds are decreasing in oxidation. Then we go down here, and we see that the number of carbon-oxygen bonds have increased more, another oxidation. And then finally we end up here, and again, the number of carbon-oxygen bonds has increased, oxidation. When we're going back this way, these are all reductions. We see the number of carbon-oxygen bonds are decreasing, while the number of carbon-hydrogen bonds are increasing. So as we go from this substance to this substance, reduction takes place. So when we go from this substance to this substance, that is a reduction. We see a decrease in the number of carbon-oxygen bonds and an increase in the number of carbon-hydrogen bonds. For the oxidation, we see a decrease in the number of carbon-hydrogen bonds and an increase in the number of carbon-oxygen bonds. So however you want to look at that, oxidation or reduction is fine. Now, obviously in the body, a steady supply of oxidizing and reducing agents must be available. Remember what I said. Many of our metabolic reactions are redox reactions. So what we have is a few what we call coenzymes that continuously cycle between their oxidized and reduced form. So what we have here is nicotinamide adenine dinucleotide as an oxidizing agent. This is the form that it's in. As a reducing agent, it looks like this with the hydrogen. I'll explain what the two hydrogens mean in a bit. Nicotinamide adenine dinucleotide phosphate. This is what it looks like as an oxidizing agent and then as a reducing agent. Flavin adenine dinucleotide. This is it as an oxidizing agent. Notice the hydrogens as a reducing agent. By the way, hydrogens in reducing agents, or I should say reducing agents, usually do have hydrogens. They provide hydride ions for the reduction reactions. Here we have flavin mononucleotide, FMN. This is the oxidizing agent form. and this is the form as a reducing agent. The reducing agents are usually going to have hydrogens associated with them. Here we can take a look at the oxidation of malate to oxaloacetate. This requires the removal of 2 hydrogen atoms. And basically what we're doing here, see this alcohol group? That's a secondary alcohol. We're going to convert the secondary alcohol to a ketone here. Okay? So how do you convert an alcohol to a ketone? Is that an oxidation or a reduction? Well, what do we know? Secondary alcohols are oxidized to ketones. So what we need then is the oxidizing agent, NAD, and then what will happen is, just like with the oxidation reaction of a secondary alcohol, I mean, that's what that is, right? What's going to happen is that hydrogen, okay, these 2 hydrogens here are going to be removed, and a double bond will form between the carbon and the oxygen, okay? And so while the oxidizing agent is used, then here the reducing agent is formed, and our oxaloacetate. This is a reaction from the citric acid cycle. So NAD plus functions as a coenzyme, and when we see these 2 hydrogens here, this hydrogen and this one, that means the 2 hydrogen atoms have been removed from the reactant. Remember, this one and this one, okay? So one has bonded to the NAD, okay? So when we write it like this, all right, one of the hydrogens is bonded to this, and then we have the slash there, and the other one is in solution as a hydrogen ion. So that's why that shows the 2 hydrogens there. So let's take a look here at the reduction of NAD+. Okay, again, remember this is our oxidized form here. So here we have the NAD+. Okay, so here's the structure for that. So what happens is the hydride ion, remember hydride, that comes from our hydride ion, looks like this. It has 2 electrons and a negative charge, okay? So we have addition of the hydride ion to the ring in the nicotinamide part of the structure, where the 2 electrons of the hydride will form a covalent bond. So again, remember, hydride ion, 2 electrons, and a negative charge. The second hydrogen atom that was removed from the oxidized substrate, okay, the substrate is the substance being oxidized, it enters the solution as hydrogen ion. The product of this reaction is often represented as this in order to show one hydrogen is bonded to the NAD and the other one is in solution. But this shows that 2 hydrogen atoms have been removed from the reactant. We already talked about step 1. We have digestion. We have step 2, the acetyl coenzyme A production. Step 3, the citric acid cycle. And step 4, ATP production. So let's move on here. Let's talk about the citric acid cycle, also called the Krebs cycle, the carboxylic acid cycle, and so on. So what happens is the carbon atoms from the first 2 stages of catabolism, those acetyl groups, are carried into the citric acid cycle. Remember, we have those acetyl groups bonded to coenzyme A. Now, remember what I told you about the phosphoryl groups and ATP molecules. When they're removed, there's a lot of energy released, okay? And that's due to that hydrolysis reaction. Well, it also turns out that the acetyl groups carried by coenzyme A are removed in an energy-releasing hydrolysis reaction. So we have acetyl coenzyme A, there is our water, okay? And we see that energy is released, in fact, quite a bit of it, negative 7.5 kilocalories per mole. So this is an energy-releasing hydrolysis reaction. Turns out that as the substance goes through several oxidation reactions, the oxidation of the two carbons to give carbon dioxide, and there's also going to be an energy transfer to the reduced coenzymes, and this takes place in about 8 steps. So we'll go through the steps in a minute. Again, this is also known as the tricarboxylic acid cycle or the Krebs cycle. Okay, so we'll talk about those 8 steps in a minute. We have this cyclic pathway, and it turns out that the product of the final step is the reactant in the first step, and that starts with oxaloacetate. Okay, so the citric acid cycle, this is a series of biochemical reactions that breaks down acetyl groups to produce energy carried by the reduced coenzymes and carbon dioxide. Again, as I said before, the acetyl groups from acetyl coenzyme A are removed through a hydrolysis reaction with a release of energy. So this is an exergonic process. This takes place in the mitochondria. 8 steps of the cycle will produce 2 molecules of carbon dioxide, 4 molecules of reduced coenzymes, and 1 energy-rich phosphate molecule. And as I said before, this is important. The final step will regenerate the reactant for step 1 for the next turn of the cycle. The cycle will operate under the following conditions. Number 1, we must have available acetyl groups from acetyl coenzyme A. Oxidizing coenzymes NAD and FAD must be available. If not, then the cycle cannot take place. Number 3, the reduced coenzymes NAD and FAD must be reoxidized via the electron transport chain, which we'll talk about in a bit. And there must be a good supply of oxygen. Why? Because the electron transport chain relies on oxygen as an electron acceptor. So this is why the citric acid cycle relies so heavily on oxygen. So, you know, during anaerobic conditions, okay, in other words, with no oxygen, then the cycle cannot take place. So plenty of oxygen is needed. Okay, so let's look at the steps of our citric acid cycle. Here are the steps. Step 1, acetyl groups enter the cycle and add to oxaloacetate to give citrate. Citrate is converted to isocitrate, which is secondary alcohol, which can be oxidized just like all secondary alcohols. Steps 3 and 4, we have oxidations that rely on NAD+ as the oxidizing agent. The electrons and energy are transferred in the reduction of NAD+. So remember, reduction, that's going to provide hydrogens and electrons. Succinyl S coenzyme A carries 4 carbon atoms to the next step, and we have 2 molecules of carbon dioxide that leave in steps 3 and 4. Step 5, it's exergonic. This is the conversion of succinyl coenzyme A to succinate. And this is coupled with phosphorylation of a phosphate molecule, guanosine diphosphate, or GDP, to give GTP. Okay, remember we're going from the diphosphate to the triphosphate. And this is an energy-rich molecule, just like ATP. And then finally in step 6, succinate is oxidized by loss of 2 hydrogens to give fumarate. Coenzyme FAD is covalently bound to its enzyme, and both participate in the electron transport chain, which we'll talk about later, by passing electrons directly into electron transport. Steps 7 and 8, cycle is complete when oxaloacetate is regenerated. So let's go ahead and take a look then at these steps. So in step 1, what we have here is acetyl coenzyme A, and in step 1, the acetyl group is transferred from acetyl coenzyme A to the oxaloacetate, which was produced in step 8, and this produces the citrate. Now in step 2, we have an isomerization reaction, and we have a transfer of the OH group to yield isocitrate. Okay, so notice the OH group was on this 3rd carbon here. Okay, there's a rearrangement. Now it's on the 4th carbon. That's an isomerization reaction. Step 3, so here's our isocitrate here, and it loses a CO2. Okay, so it loses this carbon dioxide there, and this is also oxidized. Okay, notice this carbon here. Okay, we have this OH group, so this is oxidized to, right here we see the keto group. Okay, and this is done with our oxidizing agent NAD+. Okay, or picks up the 2 hydrogens, this hydrogen and this hydrogen, and so after this oxidation, again, we end up with a molecule of carbon dioxide, and we also end up with the reduced form of NAD. Okay, and then of course our alpha ketoglutarate. So this is an oxidation just like you've seen many times for a secondary alcohol. In step 4, we see what happens here. Our alpha ketoglutarate, okay, we see another oxidation take place here. It reacts with coenzyme A, okay, and loses a CO2, so another molecule of carbon dioxide, in order to yield our succinyl coenzyme A. Okay, so that's step 4. In step 5, our succinyl coenzyme A then is kind of set free, if you will, in order to give this molecule succinate plus the coenzyme A. And this turns out to be a reaction that is coupled with the formation of GTP. Okay, so that's our high energy molecule. And then here in step 6, the succinate is, again, this is going to be oxidized. So 2 hydrogens are removed. Okay, our FAD here is our oxidizing agent. 2 hydrogens are removed from the succinate, and that yields this substance here called fumarate. Okay, and we also get 1 molecule of the reduced coenzyme. And then finally in step 7, water is added to the double bond of fumarate. Remember, we've seen that before, water being added to an alkene, and what do we end up with? An alcohol. Okay, so water is added to the double bond of the fumarate, and what we end up with here is malate. Okay, and we see that now here we have a carbon-carbon single bond, so, and the OH group. Remember, hydrogen will add to one of the double bonded carbons and the OH from another. Okay, finally in step 8, the malate, that's this substance here, this is oxidized. Again, we have NAD, so it's oxidized. So again, there are going to be 2 hydrogens removed. Okay, this is then oxidized to regenerate oxaloacetate. And of course, we have another molecule of reduced coenzyme. And then the oxaloacetate is ready then to go into the first step of another turn of the cycle. So what is the result of the citric acid cycle? Well, number 1, production of 4 reduced coenzyme molecules, 3 NADH and 1 FADH2. So 4 molecules of reduced coenzymes. Conversion of an acetyl group to 2 carbon dioxide molecules, and production of 1 energy-rich molecule, the GTP, the guanosine triphosphate. The cycle is activated when energy is needed, and it is inhibited when the energy is abundant. Next, we'll talk about the electron transport chain.